Discovery of effectors, till 2008, the phytoplasma effectors were not known. It is in 2009 that the firm reports on effectors started coming. And idea before that, it was generally believed that there are hemolysis or adhesion like proteins which are basically virulent factors, and they are responsible for the phytoplasma associated symptoms. When post-genomic era, when the whole genome sequences became available in the case of phytoplasma, then using a particular pipeline, secreted AYWB proteins, what we call SAP proteins, have been identified as the effector molecules. And the pipeline actually goes like this, that the whole of the proteome of the phytoplasma, once the genome sequence is known, is predicted. And from that whole complete proteome, those proteins which have a signal peptide at the N terminal are basically filtered, and those which have a signal P at the N terminal sites are then filtered for the presence of transmembrane domains, so that we know that all those proteins, they are actually moving towards the membrane, but because they have transmembrane domain, they cannot be secretory. So by using these two filters, the secretory proteins are identified, and in the secretory proteins, that means they are soluble proteins, they are secretory, and they are released into the extracellular environment of the phytoplasma. Then those proteins which are already known to have any function are also again ruled out. Only those hypothetical proteins which have no known functions are uh, retained, and then they are kept as potential or putative effector molecules. Then each one of them is validated by introducing these effector genes into the model plants like Arabidopsis thaliana or Nicotiana, and the transgenic plants which have now got these genes which are putative effector genes, those transgenic lines which produce symptoms which phenocopy the disease symptoms, they are validated to be the effector molecule. So this is the scheme by which effector molecules have been identified. Series of effector molecules have been identified, and their role in all the possible symptoms of phytoplasma is a very active area of research today. One of the first effector which has been identified in phytoplasma is what we call Tengu. Tengu Su is actually a symptom in one of the Japanese, as they say, about 150 years ago, Paulownia, a multi-purpose woody plant of Japan, was described to have a disease which they called as Tengu Su, which is based upon a mythical character in their mythology, ancient mythology, which is a dragon-like, goblin-like structure. And though I don't see any similarity between the symptoms and this Tengu Su, but this is how they call it. And on the basis of the scheme which I had just described, when they analyzed the phytoplasma, once the sequence was available, they found this protein which they called as a Tengu effector. So this Tengu effector is basically not a very long protein. It was formerly called a PAM765. It encodes for a 4.5 kilodalton protein, and its signal peptide region is only 32 amino acids, and rest of it is secretory. But even after reaching in the host cell, a part of it is actually cleaved interestingly, by the host machinery, and functionally only a 12 amino acid polypeptide is actually the protein which has been believed, or which is the effector molecule which actually causes the symptoms which are associated with phytoplasma associated diseases. And when the abnormal or the transgenic plants were actually phenocopying the disease symptoms, they were producing same symptoms like dwarfism or witch's broom or malformed phyllotaxis. The biochemical or the metabolomics of all these plants was further in detail studied, and it was found that they actually affect the auxin responsive factor gene family, the MIT gene family, the ARF gene family. They are the targets of this effector protein. So this is a hypothetical model which has been proposed on the basis of the effector Tengu. That Tengu is released from the phytoplasma. It is released first in the phloem, and then it reaches in the cell. And then from cell, it moves to the shoot apical meristem, and there it down regulates auxin related genes. This is a hypothetical model, and the studies are again continuing. And recently there was another Tengu like protein which has also been discovered in case of, interestingly, wheat. There's another effector molecule. So I'm just, because of the shortage of the time and limited scope of the talk, I'm just highlighting very briefly some of the important effector molecules which have been discovered and on which aggressively the work is going on today. SAP11 is another effector molecule which is actually, the gene for it is present in the PMU region. PMU region is the potential mobile unit region. Phytoplasma genome has transposon-like elements which are highly mobile, and they have multiple copies of it. And SAP11 gene is present in, it's a large transcript which has other genes also. It works like an operon, and SAP11 is one of the 5 SAPs which have been identified. And interestingly, SAP11 is more widely distributed than Tengu. It has been reported from group 1, 2, 3, 9, 10, including 12. So SAP11 has a more universal occurrence, and the literature says that there is no report so far from Phytoplasma australians. Otherwise, in many other groups, this effector has been discovered and reported. The prominent feature of it is that it has a nuclear localization signal. That means its N terminal has NLS, and once it has NLS, that means this effector protein, which is released by the phytoplasma, can straight go into the nucleus of the host cell, and that explains how it is able to hijack the genetic programming or the genes which are involved in the normal development of the host plant. Like I said, in case of Tengu, it was the auxin gene family, auxin responsive ARS gene family, which is the target of Tengu. But interestingly, SAP11's target is the TCP gene family. And now this TCP gene family, again, a very conserved and very wide gene family across all angiosperms, is a very interesting gene family, and that is totally disrupted by the SAP11 effectors. And again, as I said, this is one of the very important area, that how SAP11 is actually affecting every, one very recent paper, 2020 only, it has come that they have been able to even show that this is a paper from Saskia Hagenhaut's lab, a very outstanding work, I would say, in maize. In maize, phytoplasma is reported to be causing maize bushy stunt symptom. And the most interesting part as a scientist is that, the modern day maize is like a single stem unbranched plant, and its ancestral species from which maize has been domesticated is a teosinte maize, which is a species with many tillers, and both male and female flowers are present in good number. Once the plant gets affected by phytoplasma, it very much looks like its ancestral species from which this plant has actually evolved. So this is a very interesting study. If those of you who have not read it, I will strongly recommend it. It's interesting because when they take SAP11 from 2 different sources, one from the maize and one from Astra yellow group, and from Arabidopsis, I think they had taken it, and when they overexpress it in transgenic maize lines, the symptoms produced were different. In one case, only male flowers were produced, and in another case, only female flowers are produced. So it's a very elaborate paper, but what is important to point out here is that SAP11, that means, has its own diversity. It can actually though it has a very conserved region, that is why it can affect the TCP gene family, but within the TCP gene family also, if the SAP11 from 2 different sources is taken, it has enough divergence to produce different kind of symptoms, and that appears to be in the the carboxy end of the SAP11 effector. Coming to another effector molecule, SAP54. This is another very important and very interesting effector molecule. And the strategy for discovering it was the same, that it was passed through a pipeline. The whole predicted proteomic sequences were passed through a pipeline, and this was found to be producing, in the case of Arabidopsis, exactly the same symptoms as Phytoplasma produces. And again, now SAP54 effector is also found to be present in many groups. And so far, hello. Uh, I'm sorry. In fact, there are the same effector molecule is called biphylogen also by a different group, though, but they are one and the same thing with some sequence level difference. And all the phylogen or SAP54 groups which have been effector proteins which have been reported from different phytoplasmas has actually been even classified into different groups, and they have identified that there are 4 different phylogens of or SAP54 if you compare the sequences of the SAP54 from different regions. They have also identified what are the conserved and what are the variable regions depending upon they have done very elegant studies of deletion mutants, and they have identified the important amino acid sequences which are responsible for its function. Just like, as I said, that in case of Tengu, it was ARF is the target. In case of SAP11, it is the TCP which is the target. In case of SAP54, it is the MTF, that means the transcription factors which are containing matchbox genes. Matchbox genes are the targets of SAP54. My group, in addition to what initially I showed you, for last few years, we have been working on SAP54. And since we are working on sesame, we tried to see what is the kind of SAP54 which is present in sesame. And interestingly, we found that, again, just like it has been reported, the SAP54 which is present in sesame, which is affected by Phytoplasma, is significantly different from the SAP54 reported from other taxa. We could find as many as 13 SNPs in the sequence, and many of the SNPs were actually significant enough. That means they were non-synonymous, and they were even changing the property of the amino acid, say from hydrophilic to hydrophobic or basic to acidic. So encouraged by this information, we even tried to identify what are the interactors of the host plant, which basically are identified by the SAP54 in order to produce the symptoms. So we carried out yeast to hybrid studies by making large scale libraries, and by interaction studies using yeast-2 hybrid, and we have been able to identify at least 2 very important interactors. One is E3 ligase, and there's another one. Both the targets of SAP54 in sesame actually appear to be helping SAP54 in such a way that they are able to effectively disrupt the matchbox genes quartet, because both the interactors which we have got in sesame, they are they are constituents of the proteasome machinery, which is involved in the degradation of the proteins. So this is a model which we have proposed, that basically these interactors, they interact with the SAP54, and then they lead the SAP54 to the matchbox gene products, and that results in the breaking of this tetramer which is formed, which is a part of the ABC model of the flowering. In fact, this is one picture again in one of the paper. They had given a very interesting picture that just like we have computer malware or viruses, and they attack our computer, and because of that the whole machinery gets disrupted. So effectors can be actually compared with the computer malware, because it disrupts the normal development, and abnormal developing plants are produced. There is last but very small time I will spend in it, that nowadays we know the small RNAs have been proposed to be playing very important role in the plant development. So in fact, quite a few of microRNAs have already been proposed to be involved in the phytoplasma symptom development. So in leaf morphology alteration, in plant architecture alteration, in yellowing, in auxin synthesis, or in fact all the hormonal reprogramming, and even in the nutrient homeostasis, the role of microRNA in the phytoplasma affected plants has already been proposed, and we have also done some investigations on the microRNA profiling of the phytoplasma affected sesame plants. So where we had taken healthy plants and plant which is very badly affected by phytoplasma, and by doing the normal small RNA detection methods, we have characterized all the small RNAs which are present in healthy as well as infected plants, and we have been able to identify those microRNAs which are uniquely present in healthy or uniquely present in infected, and those which are though commonly present in both healthy and infected, but they are differentially expressed, either overexpressed in infected or underexpressed in infected, and the extent of fold change which is being observed in the host plants microRNA which are differentially expressed. So we have got some leads, we have identified some microRNAs, and we are in the process of validating the results which we have got. So this is I would like to conclude here, and I would just like to say that there are huge knowledge gaps as far as the molecular basis of symptom development is concerned. Even if we have some idea, we have no idea about the upstream and downstream processes which are affected by the phytoplasma effectors. We need to identify the vulnerable points in this effector host protein interaction, and the extent and the strength of association which is there between effector, the symptom, and the host taxa. There are several questions which need to be answered. Even for example, how the inter and intraspecific response to the same phytoplasma, same effector is being observed in nature. And it's very important to say that effector biology has this negligible work as far as perennial taxa like even herbaceous or woody species are concerned. Most of the work which is being done is validated on the model plants like Arabidopsis or Nicotiana. The woody plants or the perennial herbaceous are, you can say, an open area and where lot of work can be done. Thank you so much, sir. I'll be happy to answer the questions.